very much honored to have the privilege to introduce Taher Saif, who's our keynote presenter this morning, and he's also the winner of the Erringen Medal this year, which is awarded each year in recognition of sustained outstanding achievements in engineering science. And so I've had the great pleasure of knowing Taher for probably about 15 years. We were colleagues at University of Illinois. He's a tremendous intellect and a wonderful scientist and a really deep thinker. And he's made major contributions to engineering science over the years. And the specific citation for the medal reads as follows. He developed a novel microinstrument to study small-scale metals and embryonic neurons. He applied the instrument and discovered plastic strain recovery in nanograined metals and a fundamental link between mechanical tension in neurons and memory. And Taher has had a very impressive career. He received his BS and MS degrees in civil engineering from Bangladesh University of Engineering and Technology and Washington State University, respectively, in 1984 and 1986. He obtained his PhD degree in theoretical and applied mechanics from Cornell University in 1993. He worked as a postdoctoral associate in electrical engineering and at the National Nanofabrication Facility at Cornell University from 93 to 97. He joined the Faculty of Mechanical Science and Engineering at University of Illinois at Urbana-Champaign in 1997. And he's currently the Edward, William, and Jane Marr Gutskell Professor in this department. And he has an amazing set of interests and really incredible set of accomplishments. And I've known Taher for a long time, and we're actually collaborators. We've co-authored papers in the past, and I'm very much looking forward to his keynote presentation titled Emergent Living Robotics and Machines. Taher, it's all yours.
Thank you. Thank you very much, John, and good morning to all of you. I'm so much honored and humbled by the SES award, particularly when I look at previous awardees, and I feel truly humbled. And thanks, John, for the very kind introduction. But all the credits really go to my graduate students and postdocs over the years. They are the ones whom I have been riding on their shoulders, and in a way, I'm almost forgetting to walk. And also, my friends, my mentors who have changed my careers and my worldview, and my collaborations with you here today. Many of the students are here today, and I really cherish your trusted friendship and mentorship, both from the students as well as my colleagues and my seniors. And of course, my family and my parents, they would be really, my parents who passed away would be really happy to see me this award, and my family and my wife who have been supporting all through my career. So without further ado, I would like to introduce the topic I'm going to talk today. This is a relatively new study, new in the sense of about the last 10 years. And there are quite a few investigators involved, but I will talk about mostly the work that has been done in my lab. This is coming out from a center, NSF Center, Science and Technology Center, that is between University of Illinois, MIT, and Georgia Tech. The name of the center is Emergent Behavior of Integrated Cellular Systems, or EBICS for short. And I will describe briefly about what the center is about. The people who have really contributed are my students and postdocs, Brian Williams, Xing Tang, Piyush, Sandeep, Onur, and Jingwei Li. My collaborators for this project had been Rashid Bashir, Jun Kong, Roger Camp, Steve Stice, and Mattia Gazzola. So here is the outline. We'll take a brief look in the history of man and machine, where we are today. What are the future machines? What have we learned from our emergent living robots or machines, and some future outlook. So if we look back where we really started machines, it started from the Industrial Revolution that started from late 1700s. And some of the machines that we now in our everyday life, but of course with the new looks and new capabilities, one is the sewing machine, or making wool or cotton. for garments and textiles, and of course the steam engine that came early 1800s, locomotion taking people and materials from one place to the other. And of course, all through the 1900s, machines became much more complex, and the modern machines increased our productivity. In fact, if you look at the population growth and the GDP of the world, it turns out that after the industrial revolution, with the introduction of all kinds of machines, population increased, as well as the GDP, people's life improved. Whereas what was thought before, that as population begins to increase, there'll be a time when there'll be a competition between resources and human populations. The population cannot grow as much. But it turns out that with the introduction of the machine, we have broken the limits of Malthusian cycle, that is what is known as. So lifestyle improved with machines, with diverse applications of machines of today. And for the machines of tomorrow, we know that there are new ideas coming in research. One being the MIT's Cheetah project, where a robot with all kinds of components, but mostly done by hard materials, and then soft robots that is also more recent, came out mostly from George Whiteside's lab, where soft materials can be used to grab and move things from one place to the other. And then more recently, we have been thinking about soft electronics, wearable electronics that John Rogers has been pioneering, where the substrate is soft, deformable, and they can carry all the electronics together. More recently, maybe over the last decade or so, there have been new ideas where it is thought that the machines don't have to live only with non-living components. They could also have living components together with them. Originally started with George Whiteside's lab. The idea was, can we take a soft deformable substrate, and then can we put, in these cases, cardiomyocyte or heart cells, and if we can pattern the heart cells using some kind of patterning technique, and then if you can apply electric field, then we can create actuation. So for example, in this case, after the application of the electric field, this walker can walk on a substrate. So the idea here was patterning the cells on a substrate, applying electric field. So external influence is needed, and the patterning is involved. So in other words, assembling is involved. This is another example from Kit Parker's lab, where a stingray is being moved by applying light, in this case, again from patterned array of cardiomyocytes. So no matter which way we look at it, from past to the present, involving living and non-living, what we find is where we are today is that the machines do not self-emerge. They have to be put together. So even the life machines or the living component machines have to be organized with cardiomyocytes, patterned cardiomyocytes. So they don't self-emerge, and also they don't have consciousness. They don't have memory and learning. And that's where we asked the question about 9 years or 10 years ago, that can intelligent machines emerge from living cells? What I mean by that is it possible that we produce soft substrates, some kind of scaffold, and then we bring living cells, and the living cells interact with one another as well as with the substrate or the engineered scaffold. And through these interactions, and the word interactions is critical here, through these interactions emerge a functional biological machine. So the concept of putting cells, organizing them, patterning them is out of the picture now. We can drop them randomly. Can they emerge into a functional unit? And the cells could be neurons, muscles, endothelial cells, all that is needed. So to put the idea more into perspective, the concept is, take a group of cells, maybe engineered cells, genetically engineered cells, take a simple substrate that could be deformable. And is it possible that if we put the cells on the substrate, they would organize themselves? And then after the organization, new functions appear. And this desired function could be In my case that I'm going to show is swimming and locomotion. So in other words, can we have order to disorder transition? Can we have order from chaos? So why would we care even if we can do that? It looks like still far away. So if we could do that, then of course, in contrast to putting batteries and sensors and power sources and actuators separately, we would have all of these together within the same cell. Within the same cell, we have power source, ATP conversion, could be sensing because of the ion channels. So actuation, sensing, power source are all in one place. These machines may have autonomy, so we don't have to tell it what to do if we can reach that level. And it would be mostly highly energy efficient because that's what biological systems are. So for example, we can live with 100 watts and we do enormous amount of activity that even today's supercomputers cannot do, including image recognition, fast memory recollections, making decisions, abstract thinking. All that we do is only relying on 100 watts. So energy-wise, this would be an extremely efficient process if we can achieve these biological machines. So these are the positive sides, of course. So we started with cardiomyocytes because that's the way we thought we could create actuation. They create forces, so might be able to produce machines that would be able to do something. Now the idea is if I put cardiomyocytes randomly, then would they work at all? Because what we know from physiology is that cardiomyocytes form gap junctions or some connection between the cells through which ions move from one cell to the other. It could be cardiomyocyte to fibroblast to cardiomyocyte, but they would have to be in contact. But we don't, at this point, we were thinking of throwing them randomly without making the contacts. Would they work at all? So the question was, can cardiomyocyte interact with one another mechanically alone without gap junctions? And if so, what would be the range of interaction? So this was not known when we started the work. So at that point, to answer the first question, that can they be turned on, can they be actuated by mechanical stimulation alone? So we know that if we take living cardiomyocyte from a heart, and if we put the heart cell on a substrate by itself, then after about 24 hours, they don't beat anymore. It's not that they're dead, but they don't beat anymore. So they go into the quiescent state. So the question was that can we wake them up? So we took a tungsten probe and put the cell on a substrate, in this case, the PA gel substrate. And we moved the probe back and forth by contacting the gel, and which means that the gel gets stretched below the cell. And in that case, as the cell gets stretched back and forth, then we apply a strain on the cell. In that case, the cell gets stretched, and if that stretch could wake the cell up. So that's the idea. Now, when we do that, this is the red lines, which are the probe moving back and forth. And what we find is that the cell begins to beat after a few oscillations of the mechanical probe. Mechanical probe is not doing anything biological, nothing chemical. It's only applying a mechanical stimulation on the cell. So what we found is that the quiescent cardiac cell can be excited to beat by mechanical stimulation alone. So that was the first finding. So in this case, on the right, what you see is a probe. This is the cell that is getting stretched, and it wakes up. But then immediately that draws the following question, is that if I have a cardiomyocyte, and then there is another cardiomyocyte nearby, but no gap junctions, and if first cardiomyocyte happens to contract as a dipole, then the nearby cardiomyocyte would feel a stretch on the substrate on which it is standing, on which it is adhered. So it would get the stretch just like what we would get by the probe that I just showed. So in that case, from mechanics, we know that further these two cells would be, the less is the interaction, which dies out as inversely proportional to the distance between them, as well as inversely proportional to the stiffness of the substrate. So it is like standing on a trampoline, and if the trampoline is very soft, and there are two kids jumping on the trampoline, then one of them jumping would be felt by the neighbor. Closer they are, the more they can feel each other's jumping. The farther they are, the less they can feel each other's jumping. So that's the inverse relationship. If the trampoline is very stiff, then no matter how close, how far the two kids are, they wouldn't feel each other's presence. So that's the same way in this case. So here, the interaction is mechanical because the substrate is deformable and the distance between them. So if we stretch the argument that we can wake up a cardiomyocyte by mechanical stretch alone, then if I put two cardiomyocytes nearby, and if one of them happened to beat, then that would stimulate the neighbor, and the neighbor would wake up, and the neighbor waking up would let the previous neighbor not go to sleep, and they would go into continuous beating. So that was the hypothesis that we can draw from the simple mechanical experiment. So we put cardiomyocytes next to each other on a same soft substrate, but soft, it's 1 kilopascal gel. And these cardiomyocytes are separated from one another, but we have distance between them. And we only ask the question, then how many of the nearest pairs are beating after a few days? So the blue lines here, this is a histogram of the number of cells beating or percentage of the pairs beating together. And the x-axis is the gap between the cells. So I just want to draw your attention to the day 4 results. By day 4, when single cardiomyocyte far apart from the neighbors are known to not beat at all, but when they are within 10 microns, after 4 days, 100% of the pairs are beating. But if you go further away, none of them, there is no blue line here, none of them are beating anymore, which means what we knew before, that individual cardiomyocytes, after a day or two, stop beating, they go to the quiescent state. But if you bring them closer on this soft substrate, then they begin to wake up. So they behave like couple oscillators. When I showed the results to cardiologists and physiologists, they said, oh, you know, you might be having some gap junctions that I cannot see because they're too close, 5 micron to 10 microns. Well, as mechanicians, we know that the stiffness is also a player by which I can turn off their connections even though they're close by. So all we had to do is to do the same experiment on a stiffer substrate, 47 kilopascal versus 1 kilopascal. So I have increased the stiffness by about 50 times. So by default, I should have been closer by 50 times to have the same mechanical effect. So if I increase the stiffness and keep the distance the same, about 10 microns, then because the stiffness has gone up, mechanical communication has gone down, and hence none of these pairs are beating anymore. So this basically answers the question that being nearby, they might be forming gap junctions, which might be true, but in this case, when we bring them nearby with the same protocol, that the cardiomyocytes remain separate, with stiffness increasing, they don't beat. So do we make the point here is that cardiomyocytes, when they're close by, but not necessarily gap junctions, can communicate with one another, and they can wake up with respect to one another. So they beat as couple oscillators. To extend the idea, we put a large number of cardiomyocytes on a thin PDMS substrate, like a membrane, like a drum, and this now cardiomyocytes is coming down, and they can all beat independently. And if we now look from the top, what you would see that the whole membrane is moving in a given direction. That's because it's not perfectly isotropic, it's not perfectly axisymmetric. So there's asymmetry. So the resultant force of all the cells makes the membrane move in one direction. It's just the orthogonal way of loading a trampoline. And if we look at any single cell beating, that is the green line, and the blue line is the oscillation of the membrane that I'm going to show in the video. And what we see is that the membrane deformation or oscillation matches exactly with the oscillation of any given cell, which means all the cells have synchronized their motion, and hence the whole thing is moving in synchrony. So this is the video of the membrane moving, and you can see the contraction of a cell here contraction of a cell here, and they have all synchronized their dynamics. Now, I mentioned that the synchrony and the coordination is because of the mechanical communication. So again, a simple test would be if I cut off the membrane by a scissor, then I have disconnected this group of cells from this group of cells. And again, you see that they're out of sync now. So the synchrony and the connectivity is what drives the coupling between the cells, and that leads to the emergence of a coordinated dynamics. Now, what is the underlying principle by which this happens? We know that cardiomyocytes have stretch-sensitive ion channels. So if we put two cardiomyocytes nearby, we know each one of them is have transport of calcium in and out, as well as the internal dynamics of the calcium. Now, we also know that if the internal calcium concentration increases above a certain threshold, then there is a large influx of calcium from what is known as endoplasmic reticulum. And that influx of calcium causes the cardiomyocyte to contract, going into the systolic phase. And that calcium has to be resorbed. And that resorption time is when it goes into the diastolic phase. It relaxes. So the time constant of the beating is determined mostly by the diastolic phase. When the calcium concentration goes down below, again, this, because of the transport of calcium, the internal calcium goes up again, and then again, the heart beats or the cardiomyocyte beats. Now, if you have two neighboring cardiomyocytes, one compressed, one contracting, then the other gets stretched, and hence the stretch-sensitive ion channels in the neighbor opens up. That creates the calcium to get in from the media in which they live. So this additional calcium that is getting in is now interfering with the dynamics of the calcium inside the neighbor, and hence they begin to talk to each other. So it's the calcium transport that is being interfered by the neighboring cell. And once that happens, then they become coupled oscillators. So we can model this by adding an extra term in the well-known calcium dynamics model of the cardiomyocytes. So the term with the summation sign here accounts for the force of the dipole, the distance between the cells, and a factor that tells what is the influence of the stretch on the stretch-sensitive ion channels or transport of the calcium. So it's only this term that accounts for the crosstalk. And if we do that, and we, at least simulation-wise, if you have a large number of cardiomyocytes, and if we start them to beat randomly, then after some time, just fast forward, after some time, they begin to beat together because of the interactions, at least based on this very simple model. Now, we know that by this time, we know that these neighboring cardiomyocytes beat as coupled oscillators, and this coupling is through the deformable substrate. Deformation of the substrate by one influences the dynamics of the other one, and this crosstalk keeps them moving or keeps them beating for a long time. Their frequencies converge and they become phase locked. Now, this kind of synchrony is not unusual. There are many instances in nature, one being the firing of fireflies that has been observed for hundreds of years now, that are the fireflies in the banks of Thailand. They all fire at the same time. And also there is an interesting example. This is the case where the Millennium Bridge was opened, and soon after it was opened, lots of people were there to see the bridge. And what you will see that the bridge is shaking a little bit, but as people begin to walk, they're feeling that the bridge is shaking, but without talking to each other, you will see soon that they synchronize their motion. They begin to walk in a way as if like they have coordinated their dynamics, which of course made the bridge shaking even worse. In this case, they're not interacting by talking to each other, but they're feeling the substrate moving, and that is being felt by all of them, and they're all trying to resist it, and as a result, they all end up with this very strange synchrony. This is more classical example, the synchronization of metronomes. In this case, if you use a group of cardiomyocytes, a group of metronomes on two Coke cans, they soon synchronize their dynamics. If you take them out, they're getting out of phase because they are not able to communicate anymore through the plate, or inertial communication is turned off, and they're getting out of phase. So essentially, the point is these are oscillators, and if they have some means to communicate with one another, they become weakly coupled oscillators, and that coupling leads to the synchrony. For the cardiomyocytes, the coupling is through the mechanical interactions with one another to the substrate deformation, and they can arrive into a synchronous dynamics. So that takes out from the restriction that if they don't have gap junctions, will these cardiomyocytes work? So now we know that they would work as a single actuator, which is needed to make a biological machine. So our first goal was, can we make a machine or a locomotor or a swimmer that can go from one place to the other, something like a sperm swimming in a fluid. This is a low Reynolds number case where inertia doesn't play any role, and if the dynamic stops, then the sperm stops moving. So the idea here was that if the sperm would behave like a scallop, if it had only one hinge, like a scallop does, then it would not go anywhere, which is coined as scallop theorem by Purcell. But Purcell also gave the following problem, that if we can take two hinges, and if we have two arms like this, then one can imagine a certain sequence of these two arms, which would give rise to a net thrust. In the same lecture, he introduced the idea of a flexible oar, which is the motion of the sperm, as well as a corkscrew motion. Both of them are used by bacterial flagella. The one that you just saw here, and the other one is it keeps turning a cilia, and it can move forward. Both cases, you can get the net thrust. So our initial thought was, that can we make this kind of motion? And it was shown by Howard Stone that you can create this kind of motion, typically known as time irreversible motion, by taking magnetic beads, connecting them with the DNA, and then applying external magnetic fields, and you can create the time irreversible motion. But here you need an external magnetic field to make them swim. So we thought, how about we take a simple tail or a simple strand of a flexible material. In this case, it will be a PDMS material. We can throw cardiomyocytes on the tail and randomly, and we would know, we would expect that cardiomyocytes now would reach to a synchronous dynamics as a single actuator, and hence it would bend the tail and it can move forward. So that's the idea. So, but the fundamental thought here is they would emerge into a single actuator by communicating with one another. That's what we did. We made a long tail coated with cardiomyocytes, and once in about 3 days or so, we see this motion, which is all of them are beating together, which is what we would expect. But of course, it doesn't go anywhere. It doesn't go anywhere because we are trapped by the scallop theorem. There is no net thrust. It's like the scallop that's moving back and forth. So then we thought that maybe we need to take a closer look and mathematically model the problem. So we thought instead of coating the entire tail, how about we put the cells in one region of the tail, and that's this region. And these cardiomyocytes now would form a single actuator because of the synchronization between them through the softness of the tail. So once it becomes a single dipole, then we can work out the deformation of the tail. So this will create a moment about the neutral axis, the dipole, and this is the elastic restoring force against deformation. Y is the vertical deformation in this case, like in that I'm showing here. M is the moment applied by the dipole, and this is the drag resistance as the tail deforms in the fluid. This is the transverse drag. So this gives the force balance in the y direction. And in the x direction, we can write down the force balance, and it turns out to be there's a net force provided there is a difference between the transverse and longitudinal drag. So if I take a pen and you move along the transverse direction, then it is known that the transverse drag is twice that of the longitudinal drag. So this gives rise to a net thrust along the x direction, which would make it move. So when we do the simulation, we can show that if the cardiomyocytes are in one region, then it can move forward or it can move backward, depending on the location of the cardiomyocytes. But again, I want to remind you that this would be a group of cardiomyocytes landing on this tail. They would emerge into this synchronous dynamics or synchronous oscillation, which is making the tail turn or bend, which is giving rise to this motion. If we can do that, then we can think of optimization in the sense that we can think about what is the tail length, what is the head length that would give us the maximum speed. So now it's a little bit of on the design side, because we can predict the motion from slender body hydrodynamics. So once we have some idea that what tail length, what head length gives rise to this maximum velocity, we can make such a structure very, very simply. So now we throw cardiomyocytes only in this region, the head region and part of the tail, but not the rest of the tail. No assembly here. We are just not putting the glue in this part of the tail. Only the glue is in the head. So the cardiomyocytes land on the tail, on the head, and this part. And as expected, they all go into a synchronous contraction mode. They become a single actuator. And now, based on the simulation, if we release it from the anchor, then it should swim. And that's exactly what we see. This is speeded up by, I think, 20 times. And the swimmer is swimming as expected along its longitudinal direction. And if we look at the comparison between the theory and experiment, the red is the model. This is the location of the swimmer. The blue is the experimental observation, and experimental model is about 11 microns per second, and experimentally we find about 10 microns per second. So it's pretty close, and there is no curve fitting. There is no parameter fitting. It's the viscosity of the water and the elastic modulus of the PDMS and so on. But these cardiomyocyte-driven machines have no intelligence. There is no memory. There is no learning. It just keeps swimming forever and soon becomes boring. But it is the first evidence that we can make a group of cells landing, self-organize, emerge into a synchronous beating actuator, and that drives the motion of the machine. There is no external magnetic field. There is no external actuation. There is no placing of the cells in the right place. They themselves have placed themselves, organized their motion or dynamics. Now, can we go towards intelligent machines? So our vision was that if we can bring neurons into the picture and muscles, not the cardiomyocytes, but skeletal muscles, And then if we can have the muscles contract because the neurons are firing, just like we walk, we talk, the neurons fire, muscles contract, then we might have a swimmer that can swim when the neurons fire. So that's the idea. But there are lots of hurdles. At that point, I thought that this is never going to work because the neurons will never form junctions with the muscle, because in vivo, our neurons are extremely specific. The neuron that is innervating or forming the junction with the muscles of my finger had many other muscles along the way, but it didn't form the junction. It is very, very specific. So why would we expect the neurons randomly placed would form the junction with the muscle that's coming from randomly placed? So I thought it's not going to work. The first hurdle that the students reported is that sometimes we can form these muscles, sometimes we cannot. Now we derive the example that was done by Christian. So in Christian's lab in 2009, they did put fibroblasts in an extracellular matrix like collagen, and what happens is the cells compact the collagen and form a tissue. So here, the collagen is compacted by the cells, and this is the side view. You can see sort of a tissue being formed by the collagen. So we thought, how about we take muscle cells with collagen and matrigel, the necessary ingredient for forming the tissue, and then we can differentiate this compacted gel and cell to a myotube. But the problem was the students were saying that sometimes they form, sometimes they don't form, but they seem to have some density dependence. So we thought we explored this a little bit more deeply. Now, if we begin to sketch out this on a piece of paper, we can begin to see what might be happening. So we know that every single cell land in the collagen matrix, they begin to remodel the matrix around them. So let's say there is a zone of remodeling, which is the dotted line. If the cells are far apart, then the dotted lines would not see each other. So the cells would be doing their remodeling all by themselves, and they maintain the global symmetry. So they're not talking to each other anymore. So they are maintaining the global symmetry, and then there is no collective behavior that can arise from it. But if they begin to be closer, meaning the density goes up and the two dotted regions overlap with one another, then the symmetry is broken, because now they're beginning to see each other, and they may begin to compact the whole gel. So breaking the global symmetry would be a necessary condition for this compaction to happen, for this collective phenomenon to arise. So we did the experiments with different cell densities, and as expected, when the cell density is high, then it compacts the matrix. This is also a video where the cells are far apart, but nothing is happening. They move just a little bit, but there is no real collective behavior. What was more interesting is if we look at the cell-to-cell gap between, if you look at the gap between the cells, and if you think about the compaction, the area compaction that I'm seeing here, area compaction of the whole gel, then of course when the gap is close by, then the cell begin to compact the gel, but there is a point where about 100 micron cell-to-cell gap, there is absolutely no interactions. There is no compaction at all. We think this is like a phase transition phenomenon. There's a sharp point where compaction completely stops. This is like the Curie temperature of ferromagnetic materials, when if we increase the temperature from Curie temperature, then the magnetic dipoles begin to organize themselves and magnetization begins to happen. But above Curie temperature, this interaction between the dipoles fall apart, and the magnetization is lost. So the parallels in this case is if the cell-to-cell gap is far away, like critical temperature, Curie temperature, here the cell-to-cell gap, then the transition doesn't happen. And the measure of emergence, what is happening out of this collective phenomena, for the ferromagnetic material is the magnetization, for the cellular interaction is the compaction. So this is a typical phase transition phenomena that we think is happening because of the gap between the cells, so interaction between the cells. So we went back to Christian's paper, And we find that in his paper, what is reported is, again, the cell number per tissue against the tension that they produce. And it turns out that they have taken, they have varied the stiffness of the pillars. But what was interesting is that the data ends at this point. There is nothing below here, no data below cell number per tissue. And this data, where it ends for the two pillar stiffness, is exactly the 100 micron gap between the cells. So there must be something intrinsic for this collagen type extracellular matrix, that about 100 microns is where the cell to cell interaction falls apart. If you move further away, there is no collective behavior, and hence probably there is no data. And we are seeing the similar thing for the muscle cells. So now that we know that we need at least 100 microns between the cells, it should be closer, then we shouldn't see this sometimes happen, sometimes doesn't happen. It should be always consistent. The muscle should be forming. So we made this simple platform to understand the interaction between muscle and the neurons that would be coming soon. So we make these two pillars with the two roofs here that you can see. The idea is to fill this up with a cell extracellular matrix like collagen, the muscle cells and collagen. And again, if we are less than 100 micron between the cells, we hope that they would form, as expected, a muscle tissue that you can see from the top and from the side. These two pillars now bend, again as expected, and we can differentiate them into myotubes. And then we put a bunch of neurons here in the middle, and it turns out that these neurons begin to shoot out axons and other neurites in all directions, but soon they begin to feel the presence of the four muscles that are around them. And here is an image of the biased growth of the axons towards the muscle, and they form the junctions. And what you will see in this video, that the muscles begin to contract as the neurons begin to fire. Now, these neurons are optogenetic, so they also sense light. And once we shine the light in the middle, all the muscles begin to contract again in a synchronous fashion. So as soon as the stimulation to the neurons comes, the light shines, and then you see all the muscles are contracting synchronously, which means that all the neurons, there are thousands of neurons now, they're all firing at the same time, so that all the muscles are contracting at the same time. So after all this, we're getting back to synchrony, now not between cardiomyocytes, but between the neurons, forming many, many junctions between them and the junctions with the muscles, and firing at the same time in a synchronous way. This cyclic synchronous firing is essential for making biological machines now, because you have a control of a machine that has a cyclic oscillation. Because for low Reynolds number swimming, you need to have this cyclic actuation so that you can move forward. Otherwise, you will stop because there is no inertial effect. There is no vortex shading or anything like that. You stop the actuation, you stop motion. So the idea here now that now that we know that the neurons can form junctions with the muscle, and the muscles can form by interacting with one another. So the hope is, put the neurons, they will form the junctions, muscle will contract, and this would swim. That's exactly what we did. So this is the two tails, like the two tails that you see here. And this is the muscle strip that we form just by putting a droplet of muscle cells and extracellular matrix. They interact with one another, form the muscle strip. Neurons now begin to see that there is a muscle strip nearby, and they're progressing in a biased way through this interaction again, and they're forming the junctions between them. And once the junctions are formed, this is again optogenetic neurons. So what you will see is this is still held at the corner, and when the light shines, this is the light stimulation, and the neurons are contracting and the tails are moving. Now, once we are releasing them, once we release the swimmer, then what you'll see, and this needs So now we shine the light and the swimmer is swimming. It's swimming at a very, very slow speed. This is the location of the swimmer, and it's swimming at about 1 micron per second. So it's a really slow speed, but it is swimming. And again, if we do the simulation using the slender body hydrodynamics, like I showed before, then the motion of the experimental swimmer and the motion of the simulated swimmer, they match very, very closely. Again, there is no curve fitting because it comes from the viscosity of the fluid, the muscle contraction, which we can measure separately to predict the swimming. So what we have talked so far as a summary is that there is a crosstalk that happens at multiple hierarchical scales between the cells and between the groups of cells that leads to the emergence of these living machines. There is no patterning, there is no assembling. It's all self-assembly to the crosstalk. So when the muscle was formed, the cells talk to each other due to the proximity through mechanics to biochemistry, which you don't fully understand. They form the muscle tissue. When the neurons landed, thousands of them, they begin to talk to each other, form junctions between one another, and those neurons begin to shoot out the neurites by sensing the presence of the nearby muscle. They're about 0.5 millimeter away. It's quite far. And they form the junctions by this higher level crosstalk between them, and they form the biological machines. It's like evolution in vitro, where there could be many possibilities along the way as it develops, but eventually they end up having a same function, which is the swimming and contracting the tails. But the two machines would not be identical anymore, just like two BMWs are identical, engineered. But now we have only produced an initial condition, and that is leading towards the endpoint through the emergence at multiple hierarchical scales. So if we look a little bit closely, a little more deeply, if we ask the question that can really intelligent machines of the future emerge, can happen, we think it is possible because the neurons are getting involved now, and we know now that they can form junctions with the muscles. And if we can do that, what is the real benefit in this case is that the question of connectivity. Now we know in the electronics industry, the computer industry, the big hurdle is the connectivity. How many connections one element can have with the neighbors. And it's very, very limited, even if we go to 3D. In contrast, a neuron can have 10,000 connections from a single cell. So there's a large number of connectivity that can form. So that's a huge to the conventional computer technology. And of course, they can self-assemble, they can grow, they can heal themselves. There could be plasticity. They can learn as they go through the experiences. And of course, memory storage, computing density, power consumption, they all are in a biological domain, which is very, very efficient. So the question is, can we program these machines' behavior by conditioning and exploiting the synaptic plasticity, memory, and learning? And can we really form minds in vitro? Can they have this consciousness to the connectivity of the cells? So I'm giving you just a quick, very broad overview that if we have many, many of these neuron muscle units, then can we test that they can remember something? And we can read out by looking at the muscles contraction or the electrical activity of the neurons. So we can have three-dimensional readouts and stimulants and stimulation platforms. This is from John Rogers' lab. And in that case, we can read out what these neurons are quote unquote thinking by either from the readouts, electrical readouts or chemical readouts, as well as contraction readouts, muscle contraction readouts. And we can begin to ask the question that can it remember a pattern that it sees? Can it remember an image? Like if we expose Mona Lisa's picture few times and then take and show half the picture, do we get the same readouts? So this is the fundamental question that remains to be seen. But nevertheless, the final frontier would be that can these living machines have consciousness in the long run? And can they talk to each other with cognition, with self-awareness, because the neurons are involved? So I will end here, and thank you again for coming early this morning and hope you have enjoyed this futuristic view of where we could be. Thank you very much.
Thank you very much, Taher. That was a really wonderful talk.